Hi guys, welcome. Just setting up. Hi guys, welcome. If you have any questions, please do feel free to ask. Just thought I would jump on whilst I'm waiting on the downloading of my video from today. So if you have any questions, how are you, Chidima? Are you well? I am good, thank you. I am good. Go on, what's your question? Can you please go through the electron transport chain? Do you think it's bad to miss a practical? You are required to do 12 practicals. If you've missed a practical, which is a PAG, then yes, you will have to catch that one up. You are expected. But if it's an extra practical that you don't need to do, then it will be all right. Like I have a chromatography practical tomorrow, but there's what's competition I'm gonna miss. Oh, the competition. Well, unfortunately, the chromatography practical is really important because they either do chromatography or electrophoresis, and not many schools can do the electrophoresis. So therefore, I would say that that's what it is. Right, Hannah, you want to look at the electron transport chain. Do you want it for photosynthesis or do you want it for the electron transport chain in the mitochondria and on the cristae? Oh, okay. So if it's not the competition, I would have to say you'll have to miss the competition. Hi, Iris. Or it will be a case of you need to redo the exams, photosynthesis, of course. So Hannah, I'm on my YouTube channel as well on the Zoom. If you want to join me, I'm going to put up on the screen bits to help you to explain. So let me just get that up now. So you want the photosynthesis light-dependent reaction because you're interested in the ETC, that electron transport chain. Iris, how are you doing? Are you feeling better? Katerina, what are you confused about? Oh, I'm sorry you're having an off day. It's always the way, isn't it? Fortunately, it's never one way or the other. Okay, so Hannah, if you are still listening, I am going to now go through the electron transport chain, but you can either join me on you're on the YouTube. Perfect. Okay, so first thing to know, when we're looking at the light dependent reaction, what I always would say is make sure you understand when you talk about a photosystem what that actually is. So the photosystem is a light harvesting system and a reaction center. So what you can see here is these are all my different photosynthetic pigments. So when you look at the photosynthetic pigments, you need to understand that you have these accessory pigments, which are your xanthophylls, your carotenoids, your chlorophyll B. And then you have chlorophyll A, which is your primary pigment. And the reaction center is only made up of chlorophyll A. So what you have to understand is the accessory pigments are going to absorb light energy and they're going to transfer that to the reaction center. Now that reaction center is the one place that can become oxidized. So it's the one place it can donate its electrons to the electron transport chain. So if you understand that when a photon of light comes in and is absorbed by a pigment, its energy is transferred to the electron. That therefore excites the electron. The electron jumps up its energy level. So we've now got a higher energy level. And then what happens is as it comes back down to its original energy level, what you've then got is a release of excess energy. And that release of excess energy is necessary for the next part of the electron transport chain. So you need to know about organic cofactors, NADP, when we talk about photosynthesis, that can be reduced. So it's going to gain hydrogens. Yes, this is live if you're asking questions. It's going to gain hydrogens and electrons, and then you get reduced NADP. Going the opposite way, if we're going to lose the hydrogens and electrons, we're going to oxidize back to the NADP. So we have reduced NADP, which has gained the hydrogens and electrons, and NADP that can be oxidized. So if we're looking at non-cyclic photophosphorylation, you have that a photon of light gets absorbed, and I'm going to start with photosystem 1, okay? So a photon of light gets absorbed. The electrons from the reaction center get donated to the electron transport chain. Now, that therefore is the part up here. Now, I'm going to go back a second. This is a protein which is part of the electron transport chain, but it's also an enzyme. It is an NADP reductase. So it's an enzyme that is going to reduce NADP. So what's happened then is those, going back, electrons that were donated from the reaction center are now going to be accepted along with hydrogen ions to form reduced NADP. Now that reduced NADP, that coenzyme, is going to go off to the light independent reaction, the Calvin cycle. So the donated electrons from photosystem 2 are now up here in the reduced NADP and about to be taken to the Calvin cycle. We know that a photon of light comes in as well to photosystem 2 because photosystem is this light harvesting system with a reaction center. So that's going to absorb the wavelengths of light. That will then have the reaction center, which can also become oxidized, donate its electrons, and therefore they can make their way across from photosystem 2 to photosystem 1. So they're replacing the electrons that are currently up here. Now, as they made their way across, if I just go back a second, what you'll notice is as the electrons go across, hydrogen ions are being pumped into that thylakoid space. Hydrogen ions are charged and therefore they cannot get across the phospholipid bilayer. They are being actively pumped against the concentration gradient because they're going from a low concentration to a high concentration. So that's against the concentration gradient. And what we're getting is this accumulation of hydrogen ions in the thylakoid space, and that's why they are stacked. And so what we have is this accumulation of hydrogen ions. What will then happen is those hydrogen ions will be able to move through ATP synthase. So as they are going to go down their electrochemical gradient, they are going to move from that high concentration to that low concentration. They therefore are going to make their way down, and that will result in that energy that's needed to bring the inorganic phosphate together with the ADP. So where do the hydrogen ions come from? Well, remember, there are hydrogens and ions all over. We haven't done photolysis yet. Medicine with Sylve. Yeah, Sylve, I had to double check your name there. We're not done it yet. We're talking about just the electron transport chain before we get to photolysis. So what you have to understand, because you're asking about these hydrogen ions, my diagram's not really the right diagram. I realized this afterwards. I should really have them as hydrogen ions. But if we have taken the electrons from photosystem 2 to replace the electrons that were donated by photosystem 1. So remember, photosystem 1's electrons are up here in the reduced NADP. Photosystem 2's electrons went to photosystem 1. So we need to replace the electrons that were donated. And that's why we need to do what's called photolysis or photolysis, depending on how your teacher says it. I say it photolysis to remind myself that it's photo, light, lysis, splitting. So because of light, we are going to split the water molecules up into our oxygen and our hydrogen ions and our electrons. And therefore, the electrons can be taken back into photosystem 2. So the non-cyclic photophosphorylation, the electron transport chain, let me just add this in here. So the electron transport chain is needed to create the concentration gradient. So we have a high concentration of hydrogen ions inside so that they will diffuse down their electrochemical gradient. And as they diffuse down their electrochemical gradient, that is therefore going to give the necessary energy to bring together the inorganic phosphate and ADP to make ATP. Hannah, does that help for that one? With regards to oxidative phosphorylation is when you're talking about the cristae and you're talking about the mitochondrial process of aerobic respiration. So in oxidative phosphorylation, oxygen is the last electron acceptor, whereas in this case, the last electron acceptor in non-cyclic photophosphorylation is actually the NADP. So that's the last electron acceptor, and that's going to go off to the Calvin cycle, and it's going to then drop them off at the Calvin cycle so that they can then be used to reduce the GP into TP. Hannah, does that help with your question? Right, let me go back. Sorry, Katerina, if you're still in here, right. Coordination of the cardiac cycle and pressure changes in the heart chamber. You understand 90% of transport in animals, but the graphs in ECG confuse you so badly. So the ECG is about the electrical impulses that are going through the atria. So you've got your P wave is atrial depolarization. Your QRS complex is your ventricular depolarization. And your T wave is your ventricular repolarization. Now, you don't see the atrial repolarization because the ventricular depolarization is greater than the atrial depolarization. So that's why we have our P wave, our QRS complex, and our T wave. So when we talk about the coordination of the cardiac cycle, you're talking about the fact that the SEN is going to, it's self-excitatory because it can spontaneously generate the action potential itself, and it propagates it through the atria. Now, all the cells, you should know cardiac cells are interconnected, they're branched, and because they're branched, what that means is they all actually contract at the same time because they all get the electrical impulse at exactly the same time. So they have these like little intercalated discs which connects them together. Basically, they have proteins in the membrane which allow for the depolarization to get through at the same point. So if you think about an action potential making its way down the axon, what we're saying is with the cardiac cells, they are going to make their action potential is going to make its way across all of the atria so that they contract at the same point. And then you've got the ventricle that's going to contract at the same point as well. But you need a delay because you want it to go dum, 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 dum. And so therefore, the part between the AVN and the SEN, there is a very slow delay. So when the impulse gets passed from the SEN to the AVN slows it down to ensure that there's a boom, 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 boom. And you can actually see that delay when you look at the distance between, so you've got your P wave, the distance between the P wave and the Q, that is the delay in the AVN. So I hope that helps to answer that question. Okay, so what's the questions we have here? What's the difference between substrate level phosphorylation and non-substrate level? So substrate level phosphorylation, be careful when you say non-substrate level, because I'm like, what are you talking about? Are you talking about oxidative phosphorylation? So substrate level phosphorylation is where you're going to add a phosphate to ADP so you're transferring a phosphate to ADP from an intermediate molecule. So for example, if we're in the Krebs cycle, there is an intermediate one that you don't need to know the name of, but there's an intermediate molecule, because you know how it goes from oxaloacetate to citrate, and then citrate goes to the 4-carbon and the 5-carbon, then the 4-carbon, then the 4-carbon, then the 4-carbon. So there's so many different things in between. Thankfully, you don't learn that until you go to university. But what you have to understand is that there is substrate level phosphorylation that occurs in the Krebs cycle. That is the transfer of a phosphate to ADP to produce ATP. Now, when we talk about oxidative phosphorylation, that's where you need the oxidation reactions to happen. So you have your coenzymes, NAD, that has been reduced. You have your FAD that has been reduced. When they get to the ETC, they become oxidized. And because they become oxidized, that's why it's called oxidative, because it's a set of oxidative reactions. When one becomes oxidized, the other one becomes reduced, and so forth. So it's like a passing it on until you get to the end. And the end point is where you have your oxygen as the last electron acceptor. When that accepts the electrons and it brings the hydrogens together, it produces water. So your substrate level phosphorylation is the transfer of a phosphate from a intermediate, more reactive molecule. So the other way to look at it is also when we look at creatine phosphate. So creatine phosphate is basically the storage molecule in your muscle cells to give an immediate transfer of the phosphate to make more ATP until aerobic respiration, no, that's a lie, until anaerobic respiration takes over, and then aerobic respiration takes over. Can I go over chloride and Bohr shift? So the chloride and shift that you want to talk about, that's basically just talking about the movement of your hydrogen carbonate ions out. Let me just add in, hold on. Lots of questions tonight, guys, which is great. That's exactly, oh no, hold on. I didn't mean to do that. I must delete that slide. Okay, so if I go on to the chloride shift and we look at what happens there. So give me 1 second. I will bring in the diagram. I don't want to do that. It's transporting animals. And if you are just coming across my profile now and you've never heard of me before, guys, all of the information that I'm given, all of the course content are in playlists already in the on my YouTube channel here. So you can actually watch the playlists specifically for the topic area that you are looking for. But let's have a look at, oh no, let's try that again. So if we're looking at the chloride shift and the Bohr shift, you have to understand you've got your partial pressure. Now, partial pressure is just concentration of oxygen. So if we know that oxygen is 21%, we know the partial pressure is 21 kilopascals because we talk about it in pressure. So if we're looking at the alveoli, we know that our partial pressure of oxygen is 21 kilopascals. our partial pressure of oxygen at our normal cells is 5 to 7 kilopascals. So we can see that the partial pressure is a lot lower at normal cells than it is in the atmosphere. If your cells are very active because they've been doing lots of respiration, the oxygen, which is the last electron acceptor at the ETC, that level is going to decrease. And now remember, partial pressure is about the concentration of a gas. So therefore, when we talk about the partial pressure, the concentration of that gas has decreased because that has accepted the electrons and now being converted into water. So we know very active cells, we have more respiration therefore has a lower partial pressure of oxygen. So concentration of oxygen were to decrease, so with the partial pressure. Makes sense, hopefully. So what you should know about the oxygen dissociation curve is that the affinity for the affinity for oxygen is actually really hard at the start. To get that first oxygen molecule to be 25% saturated is really difficult. But the minute it happens, the minute the oxygen binds to the prosthetic group, the heme group on hemoglobin, what's going to happen is that's going to change the shape of the hemoglobin molecule and therefore make it easier for the oxygen to bind. And that's why you get this lag at the start. Then you have this growth part here where it's a lot easier to load the oxygen on, but then it excuse me, becomes much more difficult again when you get up to the point of being close to fully saturated. So this is about your saturation of hemoglobin with oxygen. Now, it's important to remember that I always take students back to the fact that your erythrocytes, if we were looking at your erythrocytes and what they actually have, your erythrocytes have this conjugated protein. They have 3 million hemoglobin molecules inside the cell. So the heme group, the prosthetic group, that iron is part of the heme group, which will be broken down. And that's the bit which the oxygen can bind to. So if we're saying it is fully saturated, every single one of these hemoglobin molecules has 4 molecules of oxygen attached to it and is called oxyhemoglobin. So this, when it's got 1, then 2, then 4, then 6, and then 8, it gets to this point where you can load it and then you can unload it. So the more oxygen attached, the more the affinity is increased. Wow, it's hard for me to say things when I'm going so fast. When we add things, we are adding the oxygen, we say we are loading it, that's association. When we are losing the oxygen, that is unloading, okay? So we are looking at our dissociation curve. So this is the oxygen dissociation curve. So we can see at which point it's saturated up here at 100. If it's 75% saturated, if it's 50% saturated, if it's 25. So if we're looking at a graph like this, you would want to be able to say that this actually doesn't quite hit 100% saturated before it plateaus out. That's an interesting one. But the other thing for you to note is to come down to the point in which you are seeing it on the graph at the bottom. At which partial pressure of oxygen will the hemoglobin only be 75% saturated? At which point would it be 50% saturated? And so forth. Now, I've just made this graph myself, so therefore that's not the right values that they will give you in the exam. So you want to use the graph that they have given you and those values. But what's actually happening, and this goes back to your chloride shift question, the oxygen is going to diffuse. Now, remember, you've got your alveolus, which is 1 cell thick. You've got your capillary, which is 1 cell thick. So we have a short diffusion distance. The oxygen is going to diffuse across, and the oxygen binds with the hemoglobin. Now, as you can see here, this is actually hemoglobinic acid. There is a hydrogen ion attached to the hemoglobin. So what will happen is when the oxygen binds the hemoglobin, the hydrogen ions are released. So if the hydrogen ion is released because there is the oxygen now bound, what's going to happen is here we have our shift, which at this bit is actually called the reverse chloride shift. The erythrocytes pump chloride ions out and hydrogen carbonate ions in. So if we're talking about the chloride shift, we'd be doing it the opposite way around. So because we're up at the lungs, we are doing what's known as the reverse chloride shift, because what's going to happen is that hydrogen ion and the hydrogen carbonate ion are going to come together to form carbonic acid. That can then be converted to carbon dioxide and the water molecules, and the carbon dioxide can diffuse out. So if we go down to our respiring tissue, which is where the chloride shift actually comes in, carbon dioxide comes in and reacts with water, and we have an enzyme, carbonic anhydrase. Remember, zinc is the cofactor for carbonic anhydrase. That is going to produce our carbonic acid. The carbonic acid is then going to dissociate into hydrogen ions and hydrogen carbonate ions, and the hydrogen carbonate ion is going to be pumped out of the erythrocyte. Now, the reason it's pumped out of the erythrocyte and the chloride is pumped in is because we need our cell to be maintained in its electrical chemical gradient of a resting state. So we need a similarly charged ion to be brought in as the hydrogen carbonate ion is released out. So that's why we call that the chloride shift. Chloride ion in, hydrogen carbonate ion out, therefore maintaining the resting state of the cell. If we didn't remove the hydrogen carbonate ion, because the reaction is reversible, that could come together with the hydrogen ions to produce carbonic acid. So this way, it means that we can remove the hydrogen carbonate ion into the blood plasma, and therefore the hydrogen ion is now going to bind with the hemoglobin, changing its shape, releasing the oxygen, and therefore the oxygen can now diffuse out. So therefore what you're seeing is, and this also happens, by the way, in the blood plasma, just at a slower rate, where you're going to get your carbon dioxide and water, carbonic acid, hydrogen ions and hydrogen carbonate ions. Because we've got carbonic anhydrase, the enzyme in our erythrocytes, that therefore means that this reaction happens a lot faster. But you are going to have hydrogen carbonate ions already in the blood plasma. So if we were looking at module 5 and we were looking at animal response, and we're talking about the fact that the carbon dioxide levels are increasing, because the carbon dioxide levels are increasing, that therefore means that the concentration of oxygen is being decreased because the cells are using it for aerobic respiration. They are also increasing carbon dioxide because of decarboxylation in the link reaction, decarboxylation in the Krebs cycle. Therefore, the carbon dioxide levels are increasing. That is having a big knock-on effect. Why should I ask you a question? That's a good question to ask. It could be that I could help you. It could be that I might confuse you. I don't know. It's your choice. And the other thing to note is that carbon dioxide, when it's being carried, it can be carried in different ways. So 5% of carbon dioxide is carried in the plasma. 20% of the carbon dioxide is carried as carb aminohemoglobin. connected to the hemoglobin itself, and 75% of carbon dioxide is carried as these hydrogen carbonate ions. So carbon dioxide is transported in different ways within the transport system of the animals. So if we then look at what happens when there is a high partial pressure of carbon dioxide, because the respiring tissues have been producing it, that is going to result in a lower pH. That lower pH is because of those hydrogen ions is going to promote that dissociation, the unloading of oxygen, and that is what is going to shift the dissociation curve to the right. And that's what we call the Bohr shift. So medicine with Sylv, does that answer your question about the chloride and the Bohr shift? Yes, it is okay to write just SEN in the exam. Yeah, more activity, more CO2, therefore more O2 release. Absolutely. Oh, okay, so I am teaching OCR A-level biology here for those students who have their exams in 4 weeks' time now, 5 weeks' time. So I am a qualified teacher and examiner and a former director of student progress, and I have my YouTube channel here with the whole course content. I basically realized that my students had nowhere to go to be able to learn A-level biology to a high enough level, and also for teachers who are new to the profession. So I've been teaching for over 20 years. And when I had my daughters, I needed to change the way in which I worked so that I could do what I needed to, but also still be there for them. And therefore, I decided to put my skills into learning how to do diagrams and stuff like that and helping students. So I do these free lives for any students who can't afford to pay for tuition. I offer obviously one-to-one tuition and group tuition and courses, but this is my way of helping those of you who can't afford it. Oh, very nice to meet you. Is there a way to differentiate? Let's try that again. Differentiate between the PCT and the DCT in Microsoft. Yes. So the PCT, we know what is where you're going to have selective reabsorption. And that selective reabsorption we're going to have at the PCT is of our glucose molecules. So because of that, we want all the glucose that has been removed through ultrafiltration from the bloodstream put back into the bloodstream. You want to have an increased surface area. So you're going to have lots more channel in the membrane. So therefore you would expect there to be a brush border, which is where there's lots of microvilli, increase in surface area, therefore increasing the reabsorption. When you look at the DCT won't have that, so there'll be no shaded dark part to it. Is it the 20, I want to say 19, or it's the 2020 paper that you're looking at? You should see a kind of dark shaded area on the scan, and on the other part, you just see the white lumen and then straight to the cell. That would be how you differentiate between those two. Cooperative binding, when you're talking about binding, something binding together and it being cooperative, it's that they're both working together. That bonding that's happening is together. Any advice for your exam tomorrow? Your second year uni biology. I would say second year of uni. I found that with second year, it was about being able to get the detail into my answers, but not worrying too much about it all, having the fundamentals. But I kind of scraped through first and second. I mean, no, it's a lie. First year of uni, I was really good because they gave us assessments which were completely to my style of learning. And so I aced them and didn't have to sit the actual exam because I did so well on the other work. In the second year of uni, I just scraped by. The third year of uni, I lost my nerve when it came to genetics. I had to resit the exam 3 times. Paul, the question that Iris is trying to do, they don't show the Bowman's capsule in that question. If it's the one I'm thinking about, they've given them a picture, which is an electron micrograph of the DCT and the PCT, and then asked them to explain how the structure, but they've told them that it's PCT and the DCT, but they've asked them why it's different and what makes it different. And if you don't know to look for that brush border, you wouldn't know it was there. Hi, Tony. How are you? So the glomerular filtrate rate, they do talk about the glomerular filtrate rate, and they have to be able to decipher tables of results to do with the glomerular filtrate rate, but they will give them that in the exam. They expect them to use key 2020, I knew it was one of those. They expect them to use keywords about ultrafiltration, which then means that we can work out our glomerular filtrate rate based on the creatinine that has been circulating. Oh, good. Sorry, medicine with Silva. Just seen your answer. I'm glad that helped. Right. What was the next question? Try to work my way through. Add phosphate to ADP from intermediate is substrate level. Yes, good. Can you talk about reflexes? Of course I can. What time are we at? 47. My phone will tell me it's time to go to bed soon, although I still need to send my video to my students. What am I looking for? I am looking for the reflex. So, so nervous system then, if if you are learning about the nervous system, you need to make sure that you know that we split it up into the central nervous system, which is made up of your brain and your spinal cord, and then you've got your peripheral nerves, which are all the others, which are your motor and your sensory neurons. So the key thing about a neuron is that it's going to obviously transmit a rapid electrical impulse, and they are not going to penalize you if you say action potential or electrical impulse. They seem to accept both of those answers whenever they are looking in the mark schemes. So the key thing for you to know when it comes to reflex is it is an automatic response. It is using your autonomic nervous system. So you have this stimulus, your receptors detect it, they will then send the impulse down a sensory neuron, and that will go from the receptor, to the central nervous system. You will then have these relay neurons, which are within the central nervous system. And then you'll have motor neurons that are going to conduct the nerve impulse from the central nervous system to either an effector or to a gland. Now, when you are looking at that effector is going to do whatever the response is that's being coordinated. Now, remember, this is under autonomic control. So this is an unconscious control. This is not thought of. This is a autonomic one. Sorry, I'm just trying to, thought I had another slide in here, but I didn't know to add it in. It's autonomic. So therefore, if we look here at that, you've got your nervous system, central nervous system, brain and spinal cord, peripheral nervous system, your sensory and your motor neurons. So your autonomic, subconscious control, involuntary physiological activity, parasympathetic or sympathetic when it comes to your response that you're going to do. Is it a relaxing response or is it increasing the activity? Depending on if it's the parasympathetic, it's going to release acetylcholine. If it's a sympathetic, it's noradrenaline. That's therefore going to affect the smooth muscles, glands, but also we need to add in our cardiac muscles. So that's missing here. But if we're talking about controlling the heartbeat, the SEN, and how it sends its electrical impulses, then we need to include our sinoatrial node there. The somatic nervous system is under conscious control, voluntary activity. So me doing like this all the time, that is voluntary activity. So that is using conscious control, but it's releasing acetylcholine, and that's obviously going to be affecting my skeletal muscles. So when we look at a reflex then, your reflex action is this really, really fast, only a few synapses involved. So instead of it being a sensory neuron passing the information to the relay neuron and then it going really, 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 and then back down really, 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 and then the motor neuron, what's happening is we're going to go sensory, relay, motor. Really quick response because it's a survival mechanism. So your reflex actions should be very few synapses involved and without conscious thought. So it's a case of if I put my hand to go and pick the pizza out the oven and it's too hot, what's going to happen is I'm going to automatically release the pizza base because my skin cells need to be preserved. So it's an innate genetic process that happens. I do not think about it. It is a protective mechanism. So the pathway is really simple, and we call that your reflex arc. So stimulus, receptor, sensory relay, motor, effector, response. So as I had that before, that's a very simple one. Whereas if we're talking about any of the others, then we need to put in about a whole load of relay neurons in between. So if we look here at the knee jerk reflex, then it's a spinal reflex. So if you hit just under the patella, what's going to happen is that is going to cause receptors, which are in your muscle cells, to actually detect that stimulus. That therefore means that those receptors are going to pass the information on to the sensory neuron. The sensory neuron is then going to pass the information on to a motor neuron, here to a relay neuron and to a motor neuron. And what's going to happen is you actually have two different neurons that are being activated at the same time. So the reason we do that is because we want to maintain our posture and our balance. We need to ensure that the neurons are sending the information so that we can coordinate the response. And what happens is if there is an absence, that may actually indicate a nervous problem. So if they hit under the patella, what should automatically happen is the leg should kick out without any control. And so therefore, that is showing that it is under an involuntary control. You can't stop your leg from kicking out. So therefore, it is a reflex action that is occurring. Now, if you have multiple oscillations, if you hit underneath the patella, and what then happens is the leg kicks out a number of times, that's going to indicate that there's potentially cerebellar disease because your cerebellum is the one that is actually coordinating your balance and your posture. No, the OCR biology exam doesn't start till the 5th of May for the A level. Right, okay, so is the relay neuron part of the CNS? Yes. So the relay neuron is part of your central nervous system. The peripheral nervous system is your motor and your sensory neurons. So your relay neurons are the ones which are in, or they're also called interneurons. No, 5th of June. Did I say the 5th of May? 5th of June for the biology A-level exam. And then it's the 12th and then the 18th. So it's paper 1, paper 2, and paper 3. Oh, really? Who's your chemistry teacher? That's funny. I'm now intrigued who teaches you chemistry. No way! Oh my gosh, you have to say, oh, I love Mrs. Holland. You have to say, I say hello. So that would mean you are up north then. I haven't taught with Mrs. Holland since we were um, in Huntington together. Oh wow, that's so cool. Yeah, she's an amazing chemistry teacher. She helped me so much in my first. She actually helped train me as a teacher. Oh, that's really amazing. You need to say I say hello. She's awesome. You're very lucky to have her as your A-level teacher. She is amazing. Oh, that's actually really made my day today. It's lovely when you hear things about people that you used to teach with. I'll send her a message actually to say. Which one? Which I've had so many offset lessons now. I'm like, which one did she tell you about? I can't remember. Try to think now. We're talking, I mean, that's like going back 19 years. Oh, seriously, I could not balance equations. I hated chemistry before. I had to go on like special courses because I didn't have chemistry as a GCSE. So she basically taught me chemistry, and Miss Horlock taught me the physics. I mean, biology's fine. I love biology, but the others, I was just like, what is this? Um, Tell her I've got over. You can go next time you see her, tell her I've got over my pee phobia because I have children. I had to get over my pee phobia, which was always a running joke with my students. But yeah, she did actually have to teach me balanced equations. Tell her I now I'm good at it and I can do it. Kind of. Don't put me on the spot because I probably won't be very good at it. Oh, right, guys, it is um time for my bed. I also need to send my students their lesson from today. So thank you to everyone who has joined me. I will be back on tomorrow night again. So if you have any requests, please do send them through beforehand. Obviously, if you are interested, I am doing a course on the 28th and 29th of May as a last minute intense course for those who need that just last little bit of covering everything. So it's from 9:30 till 4:30. The link will be in TikTok. The link's everywhere. I mean, it's on my website. Um, hello, Carol. Thank you very much. And yeah, thank you guys and have a good evening.